We're going to look at sensory coding in this video. How does the nervous system identify the type, strength, and location of the stimulus? So we're going to talk about how the nervous system codes for the stimulus type, the stimulus intensity, which is talking about the strength of the stimulus, and then also the stimulus location, so where the stimulus is coming from. So let's start with coding for stimulus type. So how the nervous system knows what type of stimulus the information is coming in about is one is the receptor type that is activated. So for example, the photoreceptors in the eye are going to be activated mostly by light. And then the labeled line, the pathway that's activated. So the nerve signals coming in on this labeled line from the eye, going to the occipital lobe in the back of the cerebral cortex here. The brain has labeled that pathway as being interpreted as light. So everything coming in on that labeled line is going to be interpreted as light. So if the photoreceptors get activated, which usually will be by light, then anything coming in on this labeled line will be interpreted as light. So that's how the brain will know that, hey, this stimulus that information is coming in about has got to be a light stimulus. In coding for stimulus intensity or stimulus strength, we have two things that will tell the brain about that. The first one is frequency coding, which we've learned about before. So remember, this is how frequent or how often the action potentials are coming into the cerebral cortex. So if there's a stronger stimulus, that'll create larger receptor potentials or graded potentials in the sensory neurons. And that means that the sensory neuron will stay above threshold at the first node of Ranvier and will overcome that relative refractory period, and then we can stimulate more frequent action potentials, and then the brain will then interpret that as a stronger stimulus. And then the other thing that will code for a stronger stimulus will be the number of receptors that are activated. So we call this recruitment or population coding. So if you have a stronger stimulus, that'll activate more receptors on a single afferent or sensory neuron. And then we can add together the receptor potentials, or we can activate more than one sensory neuron, and we end up increasing the frequency of action potentials to the cerebral cortex. So let's just take a look at a couple of pictures here. So in coding for the strength of the stimulus, we have frequency coding. So if we take a look at our stimulus size down here on the bottom in pink, we've got two different sizes of stimuli. We've got a smaller stimulus and then a larger stimulus. And you can see if we measure the stimulus size here in our encapsulated nerve ending, we've got our receptor potentials. We've got a smaller receptor potential with the smaller stimulus, and then we've got a larger receptor potential generated with the larger stimulus. And then if we measure the action potentials further down on the axon of the sensory neuron, you can see that with the smaller receptor potential, with the smaller stimulus that we've created, we generate less frequent action potentials than if we had a stronger stimulus generating a larger receptor potential. We stay above threshold longer, and then we'll overcome that relative refractory period, and we're going to generate more frequent action potentials. And so the brain will interpret the more frequent action potentials as a stronger stimulus. The second way that we can code for the strength of a stimulus is by recruitment or population coding. So there's two ways we can do this. So the first way involves just a single sensory neuron. So that's this picture over here on the left. So if we've got a weaker stimulus, when we push on the skin, for example, we might only activate these 4 receptors on the sensory neuron, and maybe we're going to be opening up just those ion channels there, and we can add up those receptor potentials and send some action potentials up to the cerebral cortex from there. But if we have a stronger stimulus pushing on the skin, we can actually activate more of these sensory receptors in this single sensory neuron, opening up more ion channels in each of these receptors. And then we end up creating more receptor potentials, and we can add those together. And we end up creating a much bigger receptor potential. We stay above threshold longer, and we can increase the frequency of the action potentials. And so that'll be interpreted as a stronger stimulus. And then the other way we could do recruitment or population coding is if we have several sensory neurons that are involved. So here we've got 3 sensory neurons in the skin. So with a weaker stimulus, maybe just lightly pushing on the skin, we just activate the 1 sensory neuron here in the middle, and we'd send some action potentials to the brain. But if you have a stronger stimulus where you push harder on the skin, you would also activate these other 2 sensory neurons on the side as well. And so all 3 of these would be sending action potentials to the brain. And so the brain would interpret that as, hey, this is a much bigger signal because I'm getting action potentials coming from all 3 of these sensory neurons rather than just from 1 of them. So this would also be another example of recruitment or population coding, and you're getting a signal from 3 neurons instead of just 1, and so your brain would interpret that as a stronger stimulus. Okay, and then coding for stimulus location in touch and vision, we talk about something called acuity. And I think of the word acuity as accuracy. So this is just the precision or accuracy with which you can locate where the stimulus is coming from. So this will depend on 3 different things that we're going to be talking about. So this would be important to know. So how accurate you are at locating where exactly you're being touched, or with vision, where something is located in your visual field, depends on the size of the receptive fields, the overlap of the receptive fields, and then something called lateral inhibition. So let's take a look at the size of the receptive fields first, and we'll just take a look at touch. So is it going to be easier to localize a stimulus in areas served by neurons with smaller or larger receptive fields? So we've got a sensory neuron on the left with a large receptive field, and then we've got some sensory neurons over here on the right with some small receptive fields. And so if you were to take a pencil and poke somebody's skin in the middle of this large receptive field, it would be hard to localize where exactly you were being touched in that receptive field, because you could be touching over here to the side or in the middle or on the right side. It could be anywhere in that receptive field, but it would be hard to localize exactly where. But if you have a smaller receptive field and somebody were poking you with the pencil, you've got a much smaller area to localize where you're being touched with the pencil. Okay, so it's going to be much easier to localize exactly where you're being touched if you have a smaller receptive field. And so we see this demonstrated with this 2-point discrimination test, which you do in the physiology lab. So if we take a look at a sensory neuron with a large receptive field And if you have 2 points that are a certain distance apart, and if they happen to fall in that same receptive field, you're only going to feel 1 point. You actually don't feel these as 2 separate points. So if you do that same test and you've got an area of skin with smaller receptive fields, each of these points are going to be picked up by a separate receptive field, and so you actually would feel 2 separate points. So that's 2-point discrimination. So if you have an area of skin with smaller receptive fields, you could feel the 2 points. But if you have an area of skin with a larger receptive field, you actually would not be able to feel the 2 points. It would just feel like 1 point. And if you look on the areas of the body, you can actually see how far apart those 2 points need to be before you can feel them as 2 points. So on your calf, that has the least acuity. So the 2 points have to be about 2 inches apart before you feel them as 2 separate points. And if they're closer together than 2 inches, then it just feels like 1 point. So your calf is not very sensitive. It's really hard to localize things there. But your lips are the most sensitive area on the body. So the 2 points, as long as they're a millimeter apart, you can feel them as 2 separate points. So that's the highest acuity on the body. Okay, another way that we can help determine where a touch is coming from, its location will be the overlap of the receptive fields. So for example, if you have 2 sensory neurons in the skin and their receptive fields happen to overlap, and you poke someone with a pencil right in between where those receptive fields overlap, you're going to end up stimulating both of these receptive fields and sending signals at both of these sensory neurons. And the brain will then know that, oh, you must be touching the skin right in between those 2 receptive fields. And so you would be able to localize exactly where you're being touched. And then the third way to localize where you're being touched, or this also occurs in vision, would be with what's called lateral inhibition. So this is really interesting, where the middle neuron is going to inhibit the 2 neurons on the lateral sides. Okay, so what it does is it helps make the signals from the neuron in the middle actually stand out more. And so this actually will increase the acuity and make it easier to localize where you're being touched. So let's see how this works. All right, so we've got some sensory neurons in the skin, and we've got this area, this pink circle, where we're going to be touching the skin. And notice that there's some overlap here on the sides with these 2 neurons that are kind of in blue. But we're going to be stimulating this neuron in the middle the most, okay, because we've got more of these sensory receptors in the middle that we're going to be stimulating. And so we're going to be creating more receptor potentials in this middle neuron and sending more action potentials at this middle neuron. So you can see the action potentials, the frequency of action potentials in this middle neuron is going to be much higher than in the neurons in the sides, because the neurons in the sides, we're just going to be stimulating just a couple of the receptors, because we're just going to be on the edges here, that we're going to be creating a few little graded potentials there and not sending as many action potentials up these neurons on this side. So not as many action potentials going up the neurons on the side. So these two lateral ones, not as many signals going up. And the other thing to notice is in this neuron that's going up the middle, we've got this one on the middle. This is the first order neuron, and then we've got a synapse, and then we've got the second order neuron. And we've also got some collateral axons going out to the sides, and we're going to synapse with some inner neurons. And then these inner neurons, there's an axo-axonic synapse, okay, with these blue neurons to the side. And remember, these axo-axonic synapses will affect how much neurotransmitter is released from these presynaptic neurons on the side here. So we're going to have presynaptic inhibition going on to both of these neurons that are in blue. And so when this pink neuron has action potentials going on and communicating to these inhibitory neurons to the side, they're going to be sending signals to decrease the amount of neurotransmitter that's released by these blue neurons to the lateral sides. And so they're going to decrease the amount of action potentials that they can transmit up these second order neurons. So if you look at the graphs here, we're going to send signals up this neuron in the middle, but the neurons on the side here, notice that we're going to decrease the signals by the time we get up here. And so we're going to inhibit these neurons on the side. Okay, so that's lateral inhibition. And so we end up making the signals in the middle really stand out. And so that'll allow you to really localize that poke on the skin to the middle here, because that's where you're pushing the most, is in the middle, and really make that stand out. And so that'll allow you to localize where you're being poked a lot better right in the middle. So that's lateral inhibition. Okay, and then just localizing smell and sound, because they don't have any receptive fields, but you do have two ears and two nostrils. So they'll use the timing of like when the sound hits one ear versus the other, and when the smell hits one nostril versus the other. And that'll allow you to tell like which direction the sound is coming from or which direction the smell is coming from. And so if you know of anyone who is deaf in one ear, for example, they have a hard time telling which direction the sound is coming from, because they only have the one ear working and they don't have the other ear to help them tell, you know, which ear is the sound hitting first. And so they a lot of times will turn the wrong direction, you know, that the sound is coming from.